Hello everyone. Today, for my group, I'll be presenting on biotechnology applications for mycotoxins mitigation in grains. In this presentation, I will explain what mycotoxins are, how they contaminate grains, and how biotechnology can be used to reduce this problem through biological control. So first, what are mycotoxins? Mycotoxins are toxic substances produced by certain fungi that grow on crops such as maize, wheat, and other grains. According to WHO, people can be exposed to these toxins either by consuming contaminated food directly or through animal products from livestock that have eaten contaminated feed. Some of the major mycotoxins include aflatoxins, deoxynivalenol, fumonisins, and ochratoxins. These toxins usually develop during pre-harvest and post-harvest storage, especially when grains are stored under warm and humid conditions. Long-term exposure can cause serious health problems. So, how the contamination actually occurs? Contamination can occur due to poor storage condition and management, where grains are kept in high humidity and warm temperatures. This environment allows Aspergillus flavus to grow rapidly as the fungus consumes the nutrients within the grain and release harmful aflatoxins. When the contaminated grain enter the food chain of animal and human, it will expose them to health risks such as liver damage and cause decreasing of crop value. To overcome the problem, we propose a biotechnology solution which is introducing a toxigenic Aspergillus level strands as it is incapable of producing harmful aflatoxins. These beneficial strains are introduced directly into fields or storage environments. They work via a mechanism called competitive exclusion, where they actively outcompete and displace the toxic strains for vital space and nutrients. By utilizing beneficial microorganisms to manage pathogens sustainably, we permanently halt the toxin synthesis pathway, offering a much safer, eco-friendly alternative to synthetic chemical pesticides through the biocontrol principle. Now let's look at how ectosigenic aspergillus levels works to reduce aflatoxin contamination. This method is based on a principle called competitive exclusion. Ectosigenic strains are naturally unable to produce aflatoxins. When these beneficial strains are introduced into grain fields or storage environments, they compete directly with the toxin producing strains for nutrients and space. Because the ectosigenic strains are better established, they can dominate the environment and limit the growth of the toxin strains. As a result, less aflatoxin is produced and contamination levels are significantly reduced. Studies have shown that this biocontrol strategy can reduce aflatoxin contamination by up to 80 to 90%. This help produce safer grains while reducing the need for chemical treatments. There are several benefits of using ectoxygenic Aspergillus flavus as a biocontrol agent. First, it is environmentally friendly because it relies on naturally occurring microorganisms rather than chemical fungicides. Second, it is a sustainable and cost-effective approach for farmers and grain producers. In addition, it improves food safety by reducing aflatoxin levels and protecting consumers from potential health risks. It also helps reduce post-harvest losses and economic damage caused by contaminated grains. In conclusion, aflatoxin contamination remains a major food safety concern in grain storage. However, biotechnology offers an effective solution through the use of ectosigenic Aspergillus flavus. By preventing the growth of toxin-producing fungi, this biocontrol strategy helps ensure safer grain storage and contributes to a more secure food supply. That's all from us. Thank you.